Recently, the first gene editing therapy has been approved for use in patients for the first time in a therapy for sickle cell anemia. Joining us today is Matthew Cobb, the Professor of Zoology at Manchester University, who has just written a book about genetic engineering, its history, and what it means for our future. Welcome to Future Imperfect. Hello, listeners. Welcome to another edition of the Future Imperfect podcast. This is me, Jason Kingsley. I have with me a Professor of Zoology, Professor Matthew Cobb. Would you introduce yourself, please, and maybe talk about the book that you've got, which I think is really fascinating and is going to be a theme for which we're going to chat for the next few minutes.
Hi, my name is Professor Matthew Cobb. I'm from the University of Manchester, and my title is indeed Professor of Zoology, and I do teach students about all sorts of squidgy, strange animals that they've never heard about. But that's not what my book's about. My book is actually about the history, the past, present, and future of genetic engineering. It's got two titles, which is very useful. In the UK and in the old empire, so the Commonwealth, or whatever it's called these days, it's called The Genetic Age, Our Perilous Quest to Edit Life. In the US, it is called As Gods, A Moral History of the Genetic Age. So you can kind of see there's a bit of a difference in what the two publishers were going for. And the book does both things. It is both informative and also alarming, because we're currently at the 50th anniversary of the first genetic engineering experiments. That is the ability to precisely change at will DNA in a particular organism in a particular way. We've been changing genes since we arrived on the planet, in that we have predated, we've interacted with animals and plants and microbes, and have deliberately or inadvertently altered them. In particular, once we started domestication, then we'd do artificial selection. We'd choose those horses that were stronger, or those dogs that were a bit less yappy and were kind of nicer. And that inevitably led to changes in their genomes. But of course, we didn't know what we were doing. And it was only really in the 1970s, as I say, that we developed this ability, first just with microbes, to be able to precisely alter DNA at will. And what's striking is that over the last 50 years, this has led to some amazing changes. So, for example, any listeners who use insulin, that insulin is made in a genetically engineered microbe. That is the only way. It's the best insulin you can get. The old way used to be sort of mushing up pancreases of animals that had been killed for meat, and then you'd extract animal insulin, which isn't exactly the same as human insulin. So genetic engineering has enabled us to produce this exact same stuff. And that has clearly been of immense value to people all over the world. It produces a reliable, safe supply of exactly the right drug. On the other hand, there have been repeated worries and concerns about how the technique might lead to disaster through the creation of new diseases, the possibility of deliberately creating germ warfare using genetic engineering, and most recently, the concerns over our ability now to manipulate the human genome, not just, say, in damaged red blood cells, but also to change the genes of future generations by altering the genes in the embryo, which will then go on to be transmitted to the next generation. And what prompted me to write the book is that at each occasion, there's been this new development. The scientists themselves, the geneticists themselves, have made a research pause. They have said, crikey, this is terrifying. We should think about this. We should make sure if it's going to be done, it can be done safely. And that's unique to genetics. No other science has ever done that. Not even people working on the atomic bomb. They didn't actually stop work on it. They were concerned, but they didn't stop work. Geneticists have done that 4 times, most recently in 2019. So that's what I wanted to explore. I wanted to explore my fears about where we're going, because there's even more scary things coming down the pipe, but also to look at this history of kind of social responsibility in this area of science.
I think it's particularly interesting because I suppose what it does is it short circuits a sort of natural process. You know, natural history, evolution by natural selection is where the environment impacts on the variable survival rate of a population. And therefore, to put it in very simple terms, if it's good to run faster, it'll select for the faster ones on average, not necessarily, because accidents do happen. But broadly speaking, you get faster up to the mechanical efficiency of the system. But we're talking about being able to sort of truncate the time that it takes to do that and potentially leapfrog much faster, are we? So is it sort of the idea of putting in traits into living things that there's almost no way that they would have evolved by a sort of nudge, by the sort of artificial selection methodology?
Well, certainly, there's no way that a microbe would have evolved to start producing insulin, because that's not anything that they would ever have any need for. So in a way, it's actually taking our very mechanistic view of life and actually applying it. Cells are often described at school as, well, they're like a little factory and they've got these little structures that do this, and the mitochondrion is the powerhouse of the cell. And we use all these metaphors, but that's actually what we have done. We have turned bits of life, microbial life in particular, into little factories. They can make stuff for us, and they are doing this. And as I say, loads of drugs are now produced in this way through the smartness of genetic engineering. However, I'm not sure that the kind of vision of amazingly strong new forms of farm animal, for example, that's probably not going to happen. And what we've primarily been able to do, for example, is to introduce completely bizarre bits of DNA into organisms in order to give them functions that we think are useful. To take one example, which is actually both fascinating and a good example. So Monsanto, the now defunct or now disappeared plant genes and chemical company, which has now been taken over, so the name no longer exists. Monsanto gets a very bad press because they did lots of bad things. They prosecuted small farmers for storing the genetic modified grain that they had been sold, all sorts of bad things. But when Monsanto began this work, creating GM crops in the late 60s, early 70s, their starting point was that they wanted to, as their head put it, the CEO put it, we wanted to get out of chemicals. Monsanto used to make AstroTurf, DDT, Agent Orange, everything that kind of encapsulates the horror of 1960s America. And they were very far-seeing. They said, look, we can't just keep on doing this. We've got to change our business model. The public won't accept that we just carry on splurging this stuff all over the place. And so they came up with this very clever idea. There is a form of naturally occurring insecticide, which is produced by bacteria, which go by the name of BT, nothing to do with your favorite telecoms company. This BT bacteria is allowed by, according to rules of organic farming, to be sprayed onto plants. And the bacteria then produce this insecticide, quite by chance, which will stop caterpillars from developing. So if a butterfly lays its eggs onto the plant, then the caterpillars all die. And what they did was to get the gene for this BT insecticide and put it into plants. And that meant that plants that were normally attacked by caterpillars, like maize, or that's corn to us, or cotton, are now protected kind of internally, naturally. This stuff doesn't do any harm to any other insect apart from caterpillars that try to eat the plant. It's not being diffused anywhere. It's inside the plant, and it certainly doesn't do humans any harm. And that has led to a reduction in around about 700 million tons of insecticide, because farmers don't need to spray to keep the caterpillars that are eating their plants, to keep them at bay. The plants are doing it themselves. So that's something which the plant almost certainly could not have evolved itself, but it's an additional advantage to the plant and to us. And it's hard to see any downside to that, given the state of our insect populations around the world, that this rather clever approach has led to a reduction in the amount of insecticide, plus CO2 that's not released because the tractors aren't being driven, the planes aren't dusting the crops or whatever.
But that sounds like a really good application of genetic engineering. but I remember reading about the concept of a terminator gene. Or is that silly? Or is that?
Well, it's not silly. I mean, in a way, it's at the heart of not the terminator gene, but the principle is at the absolute heart of the success of most of the post-war growth in agriculture, in that what you do is you produce hybrid crops, F1 hybrids. And that is, you've got two crops that are okay, and then you cross them and they are now amazing. And it's that F1 hybrid, which you also get in your roses and all sorts of, gardeners may have come across this. That F1 hybrid has got extra characters because it's a mixture of the two original parent types. The problem is, if you try breeding from that, then the genes that have come together are now dislocated and the next generation is no good. It's like having a cockapoodle. You've got the two sides of it and it's amazing. But if you then were to breed that cockapoodle with another cockapoodle, you start to get something a bit odd coming out. And so the F1 hybrid, which is transient and you can't breed from it, was the basis of all the seed companies going around the world saying, hey, we've got these astonishing plants that you can grow, but you can't keep the seed because it won't work next year. So you're going to have to buy it from us again. And farmers all over the world were quite okay with that. And then at the turn of the century, Monsanto, which already had a bad press for doing other bad things, which we can probably talk about, bought up a company. And that company had come up with the cunning idea of making the F1 hybrid not only pointless to plant, to self-seed and get its offspring because the genes wouldn't be right, but actually to make it sterile. And this was a patent that they took out. And Monsanto bought up this small company. And I don't think there's any evidence that they were actually interested in this gene because, it's not terribly complicated. And farmers were used to doing this anyway. And why would you want to do it? It was picked up on by opponents of GM crops who cunningly labeled it the Terminator. And so everybody got very excited about it. Not one Terminator plant has ever been created, not even in the laboratory.
Right. Okay. I can understand why it sounds awful. And it was then labeled a very catchy name.
Yeah. But as I say, it's actually what happens. Farmers all over the world were quite okay with that approach of getting these F1 hybrids because it has produced enormous increases in productivity. But there was a great deal of sensitivity because what Monsanto had also done, and other companies at the time thought about this cunning plan and thought, oh my God, if we do that, we'll be in terrible trouble. They came up with the idea of, okay, right, we've done the business with the insecticide. That's great. Fantastic. What if we could do the same trick of increasing productivity by making plants resistant to herbicide? So you think, wait a minute, how's that going to help me if I'm growing rice or whatever? Not rice, because that's not actually been manipulated in this way for interesting reasons, which we can talk about. But let's talk about corn, maize in the US. How can that help me as a farmer? Well, you spend a lot of time as a farmer getting rid of weeds. And this means you have to have the crop quite widely spaced. And then you either dig them out. You've got people in the worst case scenario. There are humans digging it out, or you try and drill them out. There's all sorts of ways of doing it, but it's very time consuming and not effective. What if your crop was now resistant to the herbicide you're going to use? You can plant your crops closer together. You spray them all with your herbicide and hey presto, all the weeds die and your plant carries on growing. And that is what's called Roundup Ready. And this was the cunning plan that Monsanto came up with. And that is what has caused them such a lot of problems. These were the seeds. They started prosecuting people because they were keeping them because they would work in the next generation. But Monsanto said, no, we own those genes, not you. You bought the magic genes from us. You rented them. So you've got to buy them again next year. And that was the contract they made. And of course, whilst this has had an effect on reducing, it's actually increased the amount of herbicide. So rather than getting out of chemicals, Monsanto have actually produced more of this stuff. They had to find a particular chemical which would do this. And this chemical, there's a lot of argument about whether it's actually bad for humans. We can leave that to one side. I'll just note that Bayer, who now owns it, is going to stop selling this to the public in Europe at least. Farmers will still be able to use it. But if you put it anywhere near a watercourse, it is devastating because it will go into the water and then it has terrible effects on wildlife. Furthermore, both that and the cunning inherent insecticide plants, they've led to the appearance of resistant strains because, evolution is smarter than you are. And so you're in fact creating new super weeds that won't be affected by these weed killers.
Yeah, because it is metaphorically an arms race.
Yeah,
And if something can survive in those circumstances, it can do an awful lot better than its competitors who are now being killed off by that chemical. And suddenly it's almost in a monoculture. It's almost got a completely clear place
absolutely.
to proliferate.
I mean, I've interviewed people who were involved in it, very smart people, and they knew that this would happen. So they said, okay, what you've got to do is you've got to allow areas, you have kind of set aside areas around your field in which you will not spray, because there the weeds can grow and reproduce, and they're not being affected by this effectively a big selection pressure you're putting on by putting on the spray everywhere. But of course, you can only do that if you've got a huge, great big farm. If you are a smallholder, and most of the world's farmers are, this approach is absolutely useless because you cannot afford to set aside enough space because it's a huge proportion of your growing area. So what's very striking is that, there's all this ingenuity. There's no evidence, I should make clear, there's absolutely no evidence that eating these crops will do you or animals any harm. In the UK, still, even after Brexit, and in Europe, these foods do not directly enter the human food chain, but both EU and Britain do import GM grain to feed to animals. And we then, if you're carnivore, you then eat the animals. Do you no harm at all? But there is no evidence that this has actually increased productivity at all. So even from the US Department of Agriculture, which is very keen on this, the data show that there's no increase in productivity. The reason being that the crops they have designed and the way they're supposed to be used and the original strains they manipulated are only really, really happy in certain environments, like, huge, great big areas of monoculture and in particular environmental conditions. In the past, you'd have had lots and lots of different local varieties of wheat or maize or whatever that would do really well in your area and might not do so well 100 miles away. And you'd be growing that and that would be fine. But now it's the same version for everybody because it's very complicated doing this genetic manipulation. So in fact, what's happened is, the kind of huge, large scale agriculture, monoculture has been imposed as the only way that you can actually make these crops work. And the result has been that certainly in developing countries, after an initial excitement, there's now very low uptake because small farmers can't make them worthwhile. And they're not the best strains of these plants that you could conceivably get. And many farmers decide, okay, I'll take my chance because the caterpillars aren't there every year. Maybe this year it'll be fine. They'll come in 3 years' time and I'll take a hit. But I'd rather do that because overall the quality and the quantity of the crop will be better.
Yes, and I suppose you've got to think of the net benefit for all of these interventions. I remember talking to a farmer friend of mine. He was talking about certain things that they do to increase the yield. And I said, but don't all these interventions individually cost you a certain amount of money? If you just plant something and let it grow, obviously you've got risk of it failing and everything, but surely there's an optimum number of interventions to do to maximize your return. And he never really thought of it that way. And I said, but, if it's costing you £10 a hectare to do that and another £10 a hectare, but you're getting £20 per hectare extra at the end of it, surely that's no net gain for you. And and he didn't really think of that kind of grand consequence anymore because he was learning from the agro business how to do things. He hadn't really factored that in, the sort of net value overall.
Well, that is one of the issues, and the quality of the crop as well. So one of the great examples of this is Burkina Faso, which produces some of the world's best cotton. And the cotton, as cotton all over the world, is attacked by these caterpillars that munch it up. And so the government said at the beginning of the century, right, you're going to start using BT cotton, which is produced in America. An awful lot of cotton around the world is BT, and it's absolutely fine. The farmers then found they couldn't sell their cotton. It worked brilliantly. They didn't get the caterpillars, but they couldn't sell the crop because the buyers, they could buy that anywhere. What they wanted was the high quality Burkina Faso cotton. And that was just not available because they weren't growing the right version of the plant because the companies back in the US had made the crop using their local versions that, probably actually suited their experimental conditions, not actually the best crops for the USA. And so they just voted with their feet. They said, right, well, we'll take our chance with the insects. Thank you very much. We'd rather be able to sell our crop. We're better off. And so the use of BT cotton in Burkina Faso has absolutely plummeted.
So in a way, I suppose with genetic engineering, you've got to think about the appropriateness of it and also the overall outcome, growing cotton that anybody can grow, as opposed to the best cotton in the world that maybe you don't make as much of it, but is going to sell because it's a premium product. It's the same as any kind of manufacturer. Premium brands sell for the premium price for a reason. That reason might be a bit sort of made up or kind of pointless.
Yeah.
It might just be pure positioning, or it might be that it's better quality and people are willing to pay for that kind of stuff.
Yeah.
So, I mean, you touched on rice, just briefly, and I don't like leaving things hanging.
Okay.
So what's the thing about genetic manipulation of rice being harder? Or
it's not harder. It's politics. I mean, this was the thing that I found most fascinating. That in China, which of course eats a lot of rice, and it's got incredible significance culturally in terms of being their staple, and it's the symbol of the wealth of the country and well-being and so on. There's been a huge debate going on in China for the last 20 years about whether they should genetically manipulate rice. So we think of China as being very monolithic and being no debate. It is unbelievable. They have TV programs, arguing about whether they should be using GM crops, in particular rice. They produce GM cotton, loads of it, but that's not the issue. It's the rice. Members of the high ups in the Communist Party and the People's Liberation Army have huge arguments. Some of them saying it's all a Western plot. It's designed to make us sterile, and the kind of things that you can hear anywhere. It doesn't do any of those things, but there is a huge fear about actually carrying out, implementing the genetic manipulation of rice in China. And Greenpeace, amazingly, is given an astonishing amount of freedom to mobilize and organize people in China against the possibility of this happening, because they're not certain about what could be the consequences of this, not primarily ecologically, but in terms of confidence, food confidence. I mean, there've been loads of scandals in China about food security with adulteration of food in all sorts of terrible ways. But this would be even more insidious because it would have come from the government. The government would have decided this. So the US has never commercialized GM rice because they are concerned about potential exports to China and trade with China and all the rest of it. So it is a hugely political issue.
It's fascinating how food can be so incredibly culturally significant.
Well, yeah, and just compare that, right? We got people, and even probably many listeners going, well, I wouldn't fancy eating any of that, but you don't know what's in it. What's it going to do to me? That was what happened at the turn of the century when there was all the Franken food and all the scares about food, most of which had nothing to do with GM crops. But that was the thing that really got hold in Europe in particular, but also in the USA. And in Europe led to the decision not to allow GM crops directly into the human food chain. So contrast that feeling, which is still very widespread, that suspicion, with the widespread use of drugs produced in GM organisms. So we've got, I'm going to inject myself with insulin that's been made in a genetically modified microbe. Okay, that's fine. But I don't want to eat GM rice or maize or whatever. By the way, if you go to the USA and you eat any maize-based snacks or cereals, you are eating GM, because 99% of US maize is GM, and it's not going to do you any harm.
It is not the topic for this podcast about sort of risk management and the way people look at risks, but I had a friend who was a little bit concerned about certain vaccinations. Mm-hmm. And again, it's not the topic for this podcast. And he said, well, I'm not sure what's in it. And I said, I've been to parties where you're just taking all sorts of stuff off complete strangers and you don't even know what on earth you're doing. How can you? Yeah. Well, I was young then, and I was like, yeah, but there's an awful lot of studies into this stuff, and there was an awful lot of not studies into the blue, odd, weird pill that you used to take. Anyway, yeah, he's quite a powerful person now. But anyway, I won't mention his name for obvious reasons. But the other thing is, I wonder what your thoughts are on why genetic engineering, it seems to trigger people in quite extreme ways sometimes, where selective breeding, let's say, or breeding a bigger warhorse or a fatter cow or I don't know, whatever it might be, a prettier dog, doesn't seem to elicit the same kind of visceral reaction. Is it because it's hidden and sort of behind the scenes?
Yeah, and I think it's a bit more complicated. So understanding it is a bit harder, and it seems to be done. It often is done by companies, which some people would see as being nefarious by definition, i.e., they're not simply aiming to produce a better product or whatever, but they also want to make money out of it. And so people are therefore suspicious, perhaps, of motivation or of corner cutting. But I think it's the idea of something weird. I've taken a gene from a fish and I've put it in a tomato. Well, that seems pretty damn weird. And it is indeed bizarre. I mean, in my own works as a scientist, this is an astonishing tool. I can't get over quite how significant it is. I study flies, right? Drosophila, tiny little flies that are the geneticist's friends. And not I, because I'm not clever enough, but clever people have introduced genes that are from jellyfish, which will glow green if you put a fluorescent lamp on them. So you can see exactly where the gene is being expressed, the exact cell. And that expression is under the control of genes from yeast. So we got a fly with a bit of a fungus, that's yeast in it, and a bit of a jellyfish. So that's mixing up things that have been split for about 1 billion years, something like that, and sticking them together, or more in the case of the fungus, and sticking them together in the same organism. Now, that just sounds bizarre, and I can see, the listeners can't, but I can see you grinning away at this strange concoction. The flies are quite happy with it. I mean, they're as happy as any other fly. And it enables scientists to see exactly what genes are doing, how they're doing it, where to understand, to be able to manipulate in all sorts of ways experimental organisms to get to a better understanding of fundamental biology. So as a tool for scientific discovery and eventually, therefore, medical discovery, because medical discoveries are built on the basic science, this has been absolutely extraordinary over the last half century. It has transformed virtually every area of biology. We've mainly been talking about the practical applications, which is how most people come into contact with this technology and with this science. What was striking, though, is that although there were a series of public debates, for example, when the first genetic engineering experiments were done, as I said, the scientists got very concerned that they might inadvertently produce a new disease that would be terrifying. And they had a research pause, a moratorium that was announced. At this stage, there are only a few hundred people in the world who could do this. This is all in the mid-1970s. And they had a big meeting at somewhere called Asilomar, which is out in California on the coast, in February 1975. And they thrashed out not whether these experiments should be done, but how they could be done safely. So they'd never actually discussed, is this a good idea? Should we be doing this? They just said, yeah, this would be amazing, partly out of excitement, partly because they could see a way already that maybe we could make insulin in a microbe and make some money. And they did this within a few years. And so basically it was focused on the biosecurity protocols, the ways of making this safe, how to do this without the damn things getting out. This led to a big debate all over the world, in fact, but in particular in the USA and in Europe. Other countries were also involved about whether this really was safe, what safety criteria. I mean, it was an astonishing amount of argument, and books were published and there were protests and all sorts of things. By the end of the 1970s, it all went away because it was shown that you could do this stuff safely. Various things that were discovered about the way that human and animal genes are organized meant that you could put one of these into a bacteria and the bacterium would go, I don't understand what you're telling me. I'm sorry, I can't do anything with that, and just ignore it. So there were things that people were worried about that they discovered they didn't need to be worried about. And perhaps above all, there was a lot of money being made, in particular by a company called Genentech, which was the first of the big biotech startups, which was created in the same week as Apple, just down the road or down Silicon Valley from where Apple was incorporated, and went public about 4 months before Apple did in 1980 and made an incredible amount of money because they had shown that they could introduce genes that would encode human hormones like insulin, like somatostatin, which is a growth hormone, and put them into microbes, and then produce them on an industrial scale. So by the 1980s, people had started to get a bit chill about it. And it was only when it started appearing in the fields that GM plants appeared in the 1990s that people started to get worried. And a large amount of that, if listeners can remember back that far, was to do with fears that had nothing to do with GM crops, but were to do with food security, in particular mad cow disease and UK beef that had, in fact, it ended up about 150 people died of this new horrible neurodegenerative disease. It was almost certainly a consequence of eating this beef that had not been genetically manipulated in the slightest, but had not been properly treated and led to this really awful disease. And that, coupled with the development of the World Trade Organization and globalization and new rules about selling stuff, that led to huge arguments throughout the world, but in particular in Europe, over whether all this was what we wanted to do. And that led ultimately to the ban on the direct sale of GM crops in Europe.
So in a way, I think probably science fiction is partly to blame for people's kind of heightened awareness of things going in the background. But to sort of stick on that science fiction theme, what about genetic manipulation of humans, of us? Is it possible to say, I want my child to have blue eyes or to be taller yet?
Okay, so at school, let's take the blue eyes example, because Nazis like that. And they're the main people who want to do this. So at school, you probably learned that, okay, you've got a gene, and if it's recessive, then it's blue. And dominant gene is brown. And so if you've got two blue-eyed parents and their child has got brown eyes, then some kind of jiggery-pokery's gone on. You know, I remember when being taught this at school, and everybody would look round at each other and think about their parents' eyes and think, Now, that is true when you teach it at school. Is it actually true? No. So there are over 60 genes involved in human eye color. And basically, though it's pretty unlikely that two people with blue eyes can have a brown-eyed child, it is not impossible, because there are so many other variants that are floating around. 60 different genes with lots and lots of different variants. So if you wanted to be absolutely certain that your child was going to have blue eyes, you would have to edit at least 60 genes. That at the moment is not possible to do that on that scale. I mean, above all, it is not safe. So this genetic manipulation of humans has happened. It happened in 2018 when a Chinese researcher called He Jiankui did an experiment that had no point. What he did was to manipulate the embryos. Two embryos were eventually born. So we now know, in fact, there were three. At the time, he only admitted to two. So there are three children in China who have had their DNA manipulated in a particular way, as desired by Dr. He Jiankui. What he said he was trying to do was to reduce their chance of getting HIV, because there's a naturally occurring variant of a particular gene, which means that you are much less likely to get HIV. And this has been well known, and people have studied this. It's been very useful studying people who don't get HIV despite risky behavior, and then understanding what it is. That's helped us understand the disease more. But you just got to ask yourself, wait a minute. So they didn't have HIV, these children. These children were quite normal. They were going to be normal before you made them. And you then altered their genes in a particular way. So what was the unmet medical need? Because we actually know lots of ways of avoiding getting HIV. It's relatively straightforward. This has been known for kind of nearly 40 years now. Don't have unprotected sex, don't share needles, and avoid being born to a mother who's got HIV. And if you do all those things, you'll probably be okay. So there was no unmet medical need. Above all, what he did was pointless, because whilst you might reduce your chances of getting HIV, you're more likely to die of flu.
Oh.
If you've got this particular variant. There's no win-wins. It doesn't work that way, you know?
Okay.
And despite we're told that this CRISPR gene editing that he used is like a pair of molecular scissors, it's amazingly precise, etc., etc., it isn't, or it is under only certain circumstances. And in the human embryo, it is now very clear that it can go horribly wrong. And even in the crappy data that he kind of half presented before he was arrested by the Chinese government and sentenced to 3 years in jail, it's obvious that, one, he didn't introduce the mutations he wanted to. Two, the mutations he did introduce, some of them have never been seen in any other human. So all the hundreds of thousands of genomes we have sequenced of humans, not one of them has one of the mutations he introduced. Finally, in a normal state of affairs, every cell in your body has the same DNA. These girls don't. Some of their cells have the mutations, others of them don't. So this is called mosaicism. It can occur naturally and can be benign or can be really nasty. We don't know what's going to happen to those girls. The Chinese government has understandably kept it all very quiet. I mean, I don't think they should be turned into a circus or anything like that, but you'd certainly want them to be absolutely, if they're not well, to be completely supported by the government and healthcare systems and all the rest of it.
How does genetic manipulation differ from cloning, for example? I'm looking at horse cloning at the moment, sort of personal area of interest of mine with my horse breeding. And I know a lot of polo players clone their horses. I suppose it's a similar category, but it's not really related, is it?
Well, cloning is the thing that got everybody really excited. I don't know why. It was most bizarre. In the 90s, when the technology began to seem possible and it was shown that you could clone, it was Dolly the sheep was a big deal, although, mammalian clones had already been created. Why certain people got excited about it is not clear. I mean, the reason why they wanted to clone Dolly wasn't anything to do with mutton or wool. It was to use her in principle as a product, as a machine, a factory for producing drugs. So rather than putting drugs into a microbe in a big vat, you'd have them in a sheep that'd be wandering around a field, and then you'd sacrifice your sheep and extract whatever you were interested in. That didn't happen. It turned out to be far too complicated. It's not efficient. It takes, you've got to have hundreds and hundreds of these cells before one of them will eventually work. Now, I didn't know about the polo business. I know that in horse racing, it hasn't worked.
Oh, okay.
They've tried, because why wouldn't you? You got your stallion, you got your horse that's Shergar or whatever. If you could dig him up and get his DNA and, put him in a cell, hey, presto, we got the same horse. But of course, not everything is genetically determined. There is also the interaction between the embryo and the parent and the mother, and then its early life. I mean, identical twins have identical DNA at the beginning. By the end of their lives, they don't. It all mutates very, very slowly. But anyway, if you know identical twins, they are uncanny, but they're not actually identical. They're not exactly the same people. They are uncannily similar compared to any two brothers or sisters, but they are still not absolutely identical. So you're not going to be guaranteed to get exactly the same horse, even if it's got identical genes. And given there are such fine margins in horse racing, it's not actually that unless you put a nag in behind a Grand National winner, then who's going to win. But it's very hard. I mean, if you could pick winners, some people would be very rich. The only people who are rich are the bookies. So it's a mug's game, mate. Don't do it. It's interesting that cloning of horses hasn't worked. So you can get, what's her name? Barbara Streisand. One of her dogs died. And so she got another dog that is the same. But is it the same? So if you're really rich, you can get your dog cloned. If you get your cat cloned, it might not have the same color, because sometimes the coloration of cats is not simply genetically determined, especially if they're tortoise shells. So people have been very disappointed. Some very wealthy people have spent a lot of money on cloning stuff, and it hasn't looked the same. So I wouldn't do that if I were you.
This is the wonderful thing about biology, is that whilst there are bits of it we can manipulate and find out about, there's an awful lot of grey areas in biology, in my experience. I was explaining the concept of ring species to a friend who'd never really heard about them before, and the idea that you have a geographical ring, if you like, of something like gulls, and each bit can interbreed, and so is therefore a sort of species-ish. But if you go to the far ends, they overlap geographically, but they can't interbreed, so they're different. And at what stage do we say they're now different? Because they're not at any one bit of it when you look too closely. It's a bit of sort of like a fractal species.
Yeah, it's not actually true anymore.
Is it not? Okay, fine.
But
is that very old information?
This is quite recent. I used to teach this until about 5 years ago when I read something. I thought, oh my God, it's not actually true.
Okay.
But no, I mean, there's no reason why it shouldn't work.
Okay.
That you shouldn't have all those small changes. and they accumulate. But at the two ends, you've got two completely distinct species. These were in gulls in particular that went round. That's probably what you learned, the gulls going round.
Yes,
there.
I think it was.
Yeah. It turns out not to be quite the case, but it doesn't really matter, because in principle, that is absolutely possible, that you can get these small quantitative changes, which when you add them all up, turn into a big qualitative change. That is, there are now two good species that won't interbreed, even though they may be able to interbreed in every point. And all the populations kind of gradate from one into the other. I guess it's, if you imagine you've got a black and white, and you're going to shade from all those different shades of gray going around to very, very light cream. That's exactly the same principle. You look at it, you go, okay, well, they're two different colors, but at every point in here, it's all just gray.
I mean, biology is all about continua, isn't it? A lot of things work on a population level, especially in evolution. And this particular person I was explaining, I was saying, look, if you have a color chart that goes from red to blue, at some stage we can say that's blue and that's red. But you might have a different opinion of what is now going to fall into red and blue,
Absolutely.
Depending on which bit you're looking at.
Yeah. No,
and it will probably change.
Listeners may remember the dress. What color was that damn dress? Was it gold or blue? I can't remember what I thought now, but I held my views extremely strongly. I got very cross about it.
But I mean, I suppose color vision is very specialist because it involves the other colors around you and how your eyes see and everything. But that's probably a topic for another conversation.
Yeah.
Was there anything you wanted to sort of bring up towards the end of this in terms of your book and the issues that you talk about?
Yeah. So we talked about GM crops. We talked about the manipulation of humans. And I think the key thing about manipulation of humans, I mean, people who think we're going to go and live on Mars get very excited about it, but you just got to think about it. Wait a minute. Who's deciding this? Who's deciding whether the genes are going to be manipulated? It's not Elon Musk saying, I want to go and live on Mars, therefore I've got to survive, minus 100-degree temperatures and no oxygen. I'm going to have my genes manipulated. The idea involves we're going to manipulate a future individual. Well, what gives you the right to do that? I mean, it's absolutely stunning in its arrogance and its immorality. Even if you think about, well, wait a minute, what about genetic disease? We can get rid of genetic diseases. Well, at the moment in the UK, if you have genetic diseases in your family, you need to know to do anything about them. Then you and your partner can go through genetic counseling. And if it's likely that you could have a child that's affected, then you do what's called pre-implantation screening. That is, you go through IVF, which any listeners who've either been through or have got friends or family have been through is no joke. I mean, you do IVF, that's easy. No, it's not. It's really tough, and in particular on the women. And most of the people who advocate this kind of stuff are men, but women know that this is absolutely horrendous. And it can bring great joy, but it's not always guaranteed to work. Anyway, you go through IVF and then you choose the embryos that are not affected. You screen them, and the embryos that are not affected by this disease, you then introduce into the uterus. And if you're lucky, they come to term and you've got a healthy baby. So that's what we do at the moment in the UK. So genetically manipulating them, so engineering those embryos so they don't express the character that you're concerned about, that technique I've described will only not work, and you'll have to do that genetic engineering, if you've got individuals who are both what's called homozygous for a recessive gene, so they've both got two copies of the same gene, or if one individual has two copies of a dominant gene, and their offspring will have one copy of it, which will then cause a disease. And something like Huntington's disease is a dominant disease. So how many couples are there around the world that fit those two categories? Because everybody else can do pre-implantation screening. The only people who could benefit from this technique of getting rid of genetic diseases, and remember, it's not the embryos who are benefiting, it's the parents. We will be meeting the needs of those parents, the desires of those parents to have a biological, unaffected offspring. And there's no right to have children. There's no right to have healthy children. And there are lots of children who need adopting around the world. So, you might question whether we should be changing everything and taking terrible risks, I think, in order to meet these desires. But how many people around the world are there? We don't know, but the best calculations are there are a few hundred.
Oh.
And that's it.
They're tiny.
And you know where they are. You know where those people will be identified. It's not going to be in poor countries. It's going to be in the USA. So in a world in which we don't even have equal access to spectacles, we're now going to introduce this new technology, which could increase inequalities even more. Above all, is not safe. I mean, I described why it wasn't in the case of He-Jon-Ki. We now know even more that manipulation of embryos in humans can cause catastrophic loss of whole chromosomes. I mean, unbelievable damage. This has been just observed in the laboratory, of course. None of these embryos have been implanted, and I hope they never will. So it's a technology which somebody said it's a solution looking for a problem. There isn't a problem that this would solve. And no, you can't go and live on Mars this way. It's just, wear a suit if you've got to go, but you're going to die anyway. Don't even go to Mars. Send a robot.
Let's just try and end on an upbeat note here.
Oh, well, no, I'm going to frighten people anymore. Now I've got another thing. Two more things I'm going to scare people out very quickly. Firstly is malaria kills around about 600,000 people a year, most of them children under 5. If you could find a genetic tool for making the mosquitoes immune to malaria so they couldn't transmit it, or even getting rid of them, why wouldn't you do it? Well, we can now do that. We can, in fact, release genes that will multiply each generation. So rather than just diluting and disappearing, which is what normally happens unless a gene has a big selective advantage, these genes copy themselves. It is literally a genetic bomb, like an A-bomb. It causes exponential growth. These things will sweep through the population. They're called gene drives, and they are being developed for the best of all possible reasons. But what could possibly go wrong? Our knowledge of ecology is pitifully weak. So that is one potential fantastic advantage, but also very alarming. And at the moment, they are not being released, and the consensus seems to be they shouldn't be at the moment, but they are being developed. Finally, there is the delightful gain of function research, and this is stuff that's been going on since the beginning of the century, and it makes viruses even more dangerous in order to predict how they might evolve in the future. And this is not behind the COVID-19 outbreak. There's no evidence at all that they're genetically engineered. They came from bats, those viruses, almost certainly. But scientists have, for the last 2 decades, been making viruses more dangerous. So at the moment, we got this bird flu outbreak, H5N1. It's really nasty in humans, but you only get it by contact. In 2011, researchers in Holland made it transmissible through the air. As they said, we have done something really, really stupid. And they were so terrified by this that, like in 1975, they said, okay, we're going to stop doing this. But they didn't say we're going to stop doing it because it's crazy. They said we're going to stop doing it until we've got better biosecurity protocols. There are now about a dozen of these projects taking place funded by the US with the alleged suggestion that it will help us judge future pandemics. All I'll say is that the one we're living through at the moment was not altered in one jot by that research. That research did not help us in any way get an insight into how the coronavirus would and is evolving. So there's lots of things to be very wary about, very worried about. And that's why I wrote the book, because I wanted to explore these fears, see whether, like past generations in the 70s and 80s, I'd go, why am I making a fuss about this? It's okay. We can have, insulin made in a microbe. That's fantastic. Why am I protesting about this? At the moment, I'm still worried. And it's fascinating. You talked about science fiction and so on. We seem to have taken our eye off the ball, because for the last 20 years, there has been no major film, book, TV series highlighting the worries, the dangers there are associated with this kind of technology. I mean, nothing's happened. Jurassic Park was the last biggie, and the ethical issues associated that, which were summated with the phrase, your scientists spent so long thinking about whether they could, they didn't bother thinking about whether they should. We seem to have just drifted into an acceptance of this reality. And even artists who you'd expect to be, oh, that's interesting, to be kind of ringing the alarm about this. And it's something you don't see, for example, with AI. AI has been around for, since Asimov's laws. People have been worried about that. And yet we're still getting films about it. And we're still worrying about it. There are books about robots running amok and all the rest of it. So something different has happened with genetic engineering. It's kind of just become part of the background, just like people don't write books about or films about the terrors of the steam engine like they did in the Victorian age, because it was a terrifying thing. It's just accepted. The same things happened to genetic engineering, but maybe it shouldn't. Maybe we need to be a bit more alarmed about this.
Okay. What positives, what sort of bullet point positive things can we hope for in an ideal situation if the problems don't come to fruition and if we take steps to try and minimize those?
Yeah.
What can you see as a sort of positive upside to this technology?
Well, already what's happened, so the insulin and more drugs will be created that way. It's an incredibly clever and efficient, in terms of the resources it takes, way of producing new drugs. If you can persuade the bug to do it, then it is amazing. And new drugs will and are being produced this way and are being discovered this way. It will lead to the development of new crops that, because of the new ways we have of gene editing, which aren't a pair of scissors, but an awful lot more precise than the old way we had of engineering plants, and which caused such a lot of debate and still do, and doubts about their safety. The new GM manipulated crops will simply change a single letter of DNA. That's all will be altered in there. But that could lead to, for example, crops that can resist desiccation, resist drought. So in a climate change world, that is going to be incredibly important, as long as the right crops, the right strains are manipulated and it's not tied in to some large-scale agriculture ecosystem. These are actually crops that can be grown around the world. That's the challenge for the big agro companies, to actually meet the needs of farmers all around the world. It is already leading to new treatments. So rather than manipulating the whole genome, which has got dangerous, bad, and hard, people will alter particular genes that are affected in individuals. So the big exciting thing that people are interested in is sickle cell disease, which affects hundreds of thousands of people around the world, causes astonishing pain and weakness. And you can change genes in red blood cells. You extract red blood cells or their precursors from the bone marrow. You manipulate them in a test tube. You then re-inject them. And there are people who have very kindly volunteered to be involved in these experiments. They're not even clinical trials. These are experiments in the initial phases, and have had their lives transformed because they have now got normal blood cells which are producing the normal kind of hemoglobin. There are now clinical trials for this taking place. So as long as the techniques are safe, and that is a big if, as long as the techniques are safe, then manipulating genes that are damaged in parts of our bodies, so not in egg and sperm that are going to be transmitted to the next generation, if you can get access to the cells and you know exactly what the problem is, then that is going to lead to astonishing new treatments for certain genetic diseases.
Wonderful. Okay, well, that's a nice place to finish. Thank you so much for sharing that journey with us through genetics. Just a quick reminder, so your book is available.
It's available in the UK. It's called The Genetic Age. In the USA, it's not out until middle of November, and it's called As Gods.
And if people want to follow you on social media or kind of communicate?
I'm on Twitter at Matthew Cobb. That's 2 T's and 2 B's.
Wonderful. Thank you very much.
Okay, all right. Thanks a lot, Jason. Lovely to talk to you.